Thanks, John. As John mentioned in his summary, synthetic herbicides have been the weed management tool of choice in developed countries for about 70 years. By chance or design, most herbicides can be used with one or more major crops. However, the companies that are involved in discovery and commercialization of these products have based the ability to generate selective herbicides, that is, herbicides that don't cause too much damage to the crop that can be used in a crop, based on molecular target site of the herbicide. In other words, there's some chemicals that will not hit a particular molecular target site in a crop but will hit it in the weed, or rapid crop metabolism of the herbicide. Despite this, it's very common with selective herbicides to get some crop damage. This is PPO herbicide damage in soybean, which you commonly get. The crop generally grows out of it, but farmers don't like to see this, and if you get too much crop damage, it's a problem. There are several ways of making crops resistant to herbicides. One is chemical safeners, which upregulate the metabolic degradation of applied herbicide in the crop. The disadvantages of that is that you're adding another chemical to the environment, and some of these safeners work on the weed as well as a crop. So it's not a foolproof method of dealing with making crops resistant to herbicides. You can do it non-transgenically by breeding or mutation breeding, where you've changed the gene that encodes the target site for the herbicide to be resistant to the herbicide. And then transgenically, which is what this webinar is about, by making crops resistant to herbicides through the use of transgenes that impart resistance to the crop. Non-transgenic crops made resistant to herbicides by mutation breeding have been commercialized. The ones that are in gray there are some that hit the market, really didn't have much impact, and in most cases were withdrawn. The ones that are dark there that I put in the box are the imidazolone resistant crops that were made resistant by mutation breeding, and these have been a fairly good commercial success. In the future, we're having a new generation of mutation-bred soybeans resistant to sulfonylureas, rice made resistant to ACCase inhibitors, and sorghum made resistant to sulfonylureas, and these products are in the pipeline coming fairly soon. And the cost of getting these non-transgenic crops produced by mutation breeding to market is a good deal less than that for producing transgenic crops. GMOs were first introduced in the early 1990s. APHIS is the agency of USDA that handles the regulation of transgenic crops, and they issue permits and notifications for doing studies with these in the environment, and then they finally approve them, which they call deregulation. And you can see from this chart here that HT, or herbicide tolerant, which is what they call them, or herbicide resistant tolerance, has been the lion's share of the GMOs that have been regulated at some stage by APHIS. So it's a major part of the GMO market. The ones that have actually been approved by APHIS for use in the U.S. and in some cases by the Canadians in North America, you see here in this particular chart. It's a fairly long list. The first one was bromoxynil-resistant cotton, and then later canola, and then we had glufosinate resistance, glyphosate resistance, and several others. Not all of these are grown now, but these have all been approved for sale in the U.S. The bromoxynil-resistant crops disappeared, I think partly because their market share was very small, but the others have had some success and in particular glyphosate-resistant soybeans, cotton, and maize, which were introduced in the 1990s. Sugar beet, which was glyphosate-resistant sugar beet, which was introduced in 2008. It's a little darker yellow there. It's had a much larger impact on that particular crop than the soybean, cotton, and maize. A limited number of transgenes have been used to generate these herbicide-resistant crops, and most of them have been genes that produce an enzyme that degrades the herbicide, thus inactivating it. Bromoxidil-resistant crops were made resistant with a microbial degradation gene. Glufosinate-resistant crops use either the BAR or PAT gene for inactivating glufosinate. Dicamba, another microbial degradation gene. 2,4-D, another microbial degradation gene. And then, but in the case of isoxaflutol, an insensitive target site gene has been added to the crop, which makes the crop resistant to some inhibitors of hydroxyphenylpyruvate dioxygenase, the target site for isoxaflutol. For glyphosate resistance, there are only 3 genes that have been used commercially. The lion's share of them use a microbial gene, which imparts a resistant form of EPSPS, the target site gene for glyphosate. It's called the CP4 gene. Only in canola was a microbial gene for glyphosate oxidoreductase used, and that was discontinued recently. And then in some maize, the TIPS gene, which is a resistant form of EPSPS, and this is a maize gene that's been site-directed, modified to be resistant to glyphosate. It has 2 codon changes that impart resistance. If you want a historical analysis of all the genes for glufosinate and glyphosate resistance and when they were introduced, Jerry Green has a paper that just went online in the journal Pest Management Science that will detail this if you're interested in more details. The adoption rate of glyphosate-resistant or glyphosate-resistant crops has been very rapid. Here we have herbicide-resistant soybean, maize, and cotton adoption from their inception in 1996 up to last year, and you can see that over 90% of these 3 major crops in the U.S. are herbicide-resistant, and almost all of that is glyphosate resistance. For sugar beets, these data were just published by Don Morishita in a paper that's in press but online now in Pest Management Science, and you can see that at the first year of introduction, the adoption rate was about 60%. After 2 years, it was 95%, and last year it was 99%. About 60% of the sucrose sugar sold in the U.S. comes from sugar beet. So this adoption has been very great. And the reason for this, one economic analysis in 2010 found that it increased farm return by almost $600 per hectare. So it was a no-brainer for sugar beet farmers in terms of economics. If we look at globally, the total global area of biotech or GMO crops since they were introduced in the mid-1990s, it's gone up linearly pretty much, and about over 80% of the transgenic crops were herbicide resistant. Here the top line is herbicide resistant. The pink squares are stacked traits, but in almost every stacked trait case there is a herbicide resistance stacked with another herbicide resistance or herbicide resistance stacked with insecticide or insect resistance. So if you add the herbicide resistant stacked trait crops to the herbicide resistant alone, the top line there, you see it's over 80%. So it's been by far the most successful form of GMO worldwide and in the U.S. too. If we look at glyphosate use before the introduction of glyphosate, it was a fairly successful herbicide, but not used when the crop was in the field. There were ways you could use it selectively to avoid hitting the crop with it, but so it was used quite a bit. But if we look at 2014, you can see there's been a dramatic change in the use. The corn belt has almost become the glyphosate belt because of glyphosate-resistant crops. USGS has very detailed records of all pesticide use in the U.S., and you can see the influence of herbicide-resistant crops and looking at some of their charts. I mentioned bromoxynil-resistant cotton had been introduced and then left the market. Here you see the red bars are the introduction and then loss from the market of bromoxynil-resistant cotton. If we look at glyphosate use, you can see after glyphosate-resistant crops were introduced in 1996, there was a huge increase in glyphosate use. The green bars are soybeans, the yellow is corn, cotton is red. Cotton does not involve as many acres as the other two crops, but you can see its use in cotton went way up. And in about 2011, the glyphosate use flattened out, primarily because the use in these three crops had reached maximum levels at that time. And glyphosate has become the major herbicide in North America. This is data from the Economic Research Service. So why glyphosate? It's a very broad spectrum herbicide. It kills essentially all weeds, which is, it's not selective. And it translocates readily through the weed, the target plant, and acts very slowly. So it kills all the growing meristematic tips, and the plant does not come back. So it's very effective as a herbicide, much more effective than most other herbicides. And after the patent was lost in the year 2000, after the patent expired, it became very inexpensive herbicide. So it's very cost effective. And it worked much better than most other herbicides. Before glyphosate use in soybeans, not a really high value crop, farmers used a lot of herbicides and still had weeds. This was glyphosate use in glyphosate-resistant crop. At least for the first few years of glyphosate-resistant crops, the crop was essentially weed-free. Farmers really liked that. So reasons for adoption: reduced cost of weed management, improved weed management, simplified weed management, and this can't be overemphasized. Before the glyphosate-resistant crops, farmers had to use complex herbicide prescriptions depending on what weed species were in their field, and sometimes you had to hire a consultant to give you this prescription. So small farmers couldn't afford a consultant, so this was really advantageous to weekend farmers who had small acreages. They could go out and it was a no-brainer. You spray a couple of times or once and you take care of your weeds. Reduced herbicide damage to the crop and increased yields, and the bottom line was increased farm income. So naturally, over a very short period of time, adoption was extremely high. There's been some publicity that there are effects of glyphosate on herbicide-resistant crops and crop health, and the negative effects were thought to be due to glyphosate effects on mineral nutrition in glyphosate-resistant crops, and either directly or indirectly effects on plant pathogens in glyphosate-resistant crops. Glyphosate does chelate metal cations, and but so do some other compounds that are not herbicidal. There were thought to be direct effects on photosynthesis in some papers, and but the high level of resistance provided by inserting a gene for glyphosate resistance is pretty much proof that this isn't the case in in studies that have been done, the resistance factor between the isogenic lines of, I'm not sure whether this is soybean or canola, but both have been looked at, and essentially the resistance factor is 50-fold. To get phytotoxicity, you need 50-fold more glyphosate than you would need to kill a weed, and no farmer is going to spray anywhere near that amount. In a very extensive study in Journal of Ag and Food Chemistry, a group of people looked at all the data and came to the conclusion that there probably aren't any effects on yield or mineral nutrition in crops. And if we look at yield of the 3 major herbicide-resistant crops, look at the data from USDA, the yields fluctuate from year to year depending on the weather, but after introduction of herbicide-resistant crops, the shaded area in each graph, the more or less linear increase in yields continues, 1 or 2% per year for all 3 crops. Now, whether this would have occurred without glyphosate-resistant crops or whether the yield increases would have been greater, we don't know. There's no control to know the answer to that question. These data have been extended to more recent data in a recent article in Fortune magazine where they used the same data we used. You see that one big dip there, that was due to a drought, but this is the shaded areas again since the introduction of glyphosate-resistant corn and soybeans. Since 1980, there's been a 81 to 85% increase in the yield of both of those crops, even though the adoption rate of glyphosate resistance has been over 90% in each of them. And they mentioned that's despite climatic problems that have occurred during that time period. Are there environmental and toxicological issues? I'm not a toxicologist, so I'm not going to speak in detail on this. But last year, the National Academy of Sciences published a very extensive review that took them several years to do. This is online. You can go to the website that's on this slide and download the whole almost 500-page report. And just to summarize some of the things they said, the large number of experimental studies provided reasonable evidence that animals were not harmed by eating food derived from genetically engineered crop. And I remind you, over 80% of those crops are glyphosate, are herbicide resistant, mostly glyphosate resistance. Livestock health before and after introduction of genetically engineered crops showed no adverse effects associated with them, and epidemiological data on incidence of cancer and other human health problems over time found no evidence that foods from GE crops were less safe. I'm not a toxicologist, but that was their conclusion from a committee of toxicologists. We did a comparison a few years ago of the environmental effects of glyphosate-resistant crops on weed management methods, and overall there were several effects. At least during the first few years after the introduction of glyphosate-resistant crops, there was lower soil loss due to lower tillage, less herbicides in water, less fossil fuel use, largely due to less tillage, about the same pesticide use. Now, mind you, this is during the first 10 to 15 years after the introduction, and lower soil compaction and a lower environmental quotient. As I mentioned, glyphosate-resistant crops allowed farmers to eliminate tillage in many cases. These are data from Argentina, where the adoption of glyphosate-resistant crops was very much the same as in the U.S. And you can see that the use of no-till farming in soybeans went up linearly with the adoption of GM soybeans. There are similar data for the U.S. This is herbicide use from the Economic Research Service. You can see that all other herbicides in soybeans, the use went down. Glyphosate use went up with the adoption of herbicide-tolerant soybeans and corn. These are some data from a study by Sylvie Bonny in France. She looked at all the data in the U.S. for herbicide-resistant soybeans, and you can see the amount of glyphosate. The pink rectangles went up over time after the adoption in 1996. The amount of herbicides stayed about the same during her time period. These are data from the Economic Research Service on cotton, maize, and soybean herbicide use. It fluctuated, but it was about the same. No big increase or decrease in overall kilograms per hectare of herbicide, or pounds on the ground, as some people would say. However, not every herbicide is the same. The environmental risk with different herbicides varies. And in Sylvie Bonny's study, she looked at the environmental risk quotient using the Kovacs equations from Cornell University, and the environmental impact quotient went down in glyphosate-resistant soybeans in the U.S. as the adoption of glyphosate-resistant soybeans went up. Notice this only goes to 2006. So at least during the first few years of adoption, environmentally, this was very good. There are a lot more data that I don't have time to provide. Example of some of the things that happened: an arsenical herbicide, MSMA, was used commonly in cotton. You can see the red bar is cotton, until the introduction of glyphosate-resistant cotton, and you can see its use went way down after the introduction of glyphosate-resistant cotton. Similar graphs can be made for other herbicides, so that's one reason the environmental impact quotients went down generally. This is MSMA use before glyphosate-resistant cotton. This is afterward. So almost all fields look like this prior to glyphosate-resistant weeds. Life was good. Farmers were happy. But this man would have predicted you're going to have a problem sooner or later, probably sooner than later, simply because of the huge selection pressure over vast areas when you use this technology year after year. And farmers did use it year after year. It saved them money. It was simple, and it did the job better than anything that they'd ever used before. Any evolutionary biologist would have predicted, and many did, that you're going to have a problem. And sure enough, in the early years of this century, you started having things like this. This is Palmer amaranth in a cotton field. You can't see the cotton. So there were disastrous situations like this. This Palmer amaranth has evolved resistance to glyphosate. And if we look at the species that have evolved resistance to glyphosate worldwide, you can see it's gone up linearly, interestingly, since about the same time glyphosate-resistant crops were introduced. Now, not all of these evolved resistance in glyphosate-resistant crops, but certainly glyphosate-resistant crops were responsible for the evolution of resistance to quite a few of these species. And we had multiple cases of the same species evolving resistance to glyphosate in different parts of the U.S. where glyphosate-resistant crops were heavily used. So it's become a big problem. These weeds have found, I'll say, weeds do not evolve resistance to glyphosate very easily. It's not a one codon change that causes a resistance. So it has to be a fairly complicated mechanism of resistance. In some cases, in these species, for example, you have gene amplification where you have multiple copies of the target site which dilute out the herbicide. That's one mechanism. In some cases, you have multiple mechanisms. Another case, in the case of giant ragweed, the actual molecular means of how this occurs, we don't understand, but some people call it the Phoenix type glyphosate resistance, where you spray the weed, it dies very quickly instead of very slowly, as you see the sensitive plant does on the left. And the sensitive plant dies slowly, but it's completely killed. But after several weeks of treatment, the resistant one, after 24 hours, you can see you have very rapid damage. Those leaves that are first hit with the herbicide die, but the other parts of the plant grow back. Here we have a 2-codon change in EPSBS, similar to what we have with the TIPS gene for crop resistance that has occurred in goosegrass. And you can see the back row back there with the TIPS mutation with the 2-codon change is extremely resistant to glyphosate. So there are several mechanisms that weeds have found to become resistant to glyphosate. It's a problem, and it costs farmers money. So the economic returns that they got early on with this technology have been eroded quite a bit. This is data from Sosnowski and Culpepper in Georgia a few years ago. Herbicide use increased from 5 to 12 kilograms per hectare in some cotton fields where they had large amounts of glyphosate-resistant Palmer amaranth. If you want to know more about the economics of glyphosate resistance, ARS has this fairly recent report that is available online, and a very recent paper that I was informed of. This is actually published in 2017, but it's an analysis of 2012, mainly glyphosate resistance to cotton farmers, and it increased their cost of weed management by $85 to $135 per hectare in the southeast of the U.S. in that year, and things have generally gotten worse. So another fine mess we've gotten into by not paying attention to Charlie Darwin. There are some solutions, at least short-term solutions. We have deregulated stacked herbicide-resistant crops that have, in addition to glyphosate resistance, or instead of glyphosate resistance, have resistance to other herbicides stacked with them, and this will allow farmers to diversify the herbicides they're using and buy them a little time. But these are old herbicides that we're making the crops resistant to, and these herbicides like 2,4-D have been used for 70 years. You can see they've been used continually for many years. This is, again, is USGS data. Dicamba has been used for many years, and for these next generation herbicide-resistant crops, we already have weed species resistant to them, and we'll probably get more if we overuse these new products that are coming online. You can see that the beginnings of glyphosate-resistant weeds and glyphosate-resistant crops have increased. Glufosinate use in glufosinate-resistant crops, you can see since 2010, especially in wheat and cotton, glufosinate use has increased significantly. What's next that is specifically related to glyphosate? At least one company is using RNAi, sprayable RNAi targeted to EPSBS. Weeds that have evolved resistance to the herbicide would not be resistant to the RNAi, so this can be used to kill glyphosate-resistant weeds. That may or may not come online, but it's one solution. As the cost of RNAi goes down, it could be a commercial feasibility. There are other glyphosate-resistant genes that are patented and people have proposed, but these are not going to deal with herbicide-resistant crops or weeds, so it's unlikely that these will be marketed. We have patents on genes for resistant to almost every herbicide class now. Here's an example of some of them. With some of these herbicides, resistance does not evolve very easily. For instance, PDS phytoene desaturase. And there are patents for genes for resistance to this. So some of these could be useful in the future. Companies have, as I said, patented these, and in some cases almost got them to the market. This is a butylphenol resistant corn that you can see. This was published over 10 years ago by a company. You can see the resistant crop is quite resistant versus the wild type, but it was decided not to market this at the time. But these could come online in the future. I mentioned phytoene desaturase inhibitors. An aquatic weed, hydrilla, evolved resistance to fluordone, one of these phytoene desaturase inhibitors. This is the resistant hydrilla. The one on the left is bleached by the herbicide, and we pulled the gene out of hydrilla, and in a heterozygous transformed Arabidopsis you get a certain level of resistance, and in the homozygous you get complete resistance. If you look at the resistance factor for fluordone, it's about 10-fold, but for norfluozine, another chemical analog of fluordone, it's 50-fold, which is the same level of resistance that you get with glyphosate-resistant crops. What's interesting is you get negative cross resistance to some other phytoene desaturase inhibitors. There are ways of working that into resistance management schemes. So, and as I mentioned, this is a herbicide class that there's only one weed that's actually evolved target site resistance to it, and that is hydrilla. There are no terrestrial weeds with resistance to it, and we know that in many cases the herbicide, the weeds that are out there need 2 codon changes to have a herbicide-resistant phytoene desaturase, whereas hydrilla only needed 1 codon change. So the chances of target site resistance are very low. So there are some possibilities there. If we look at GMO permits and notifications over time, you can see that it's not really growing. The cost of getting a GMO to market is very high. So companies have to be very selective about what they're going to try to market, and there are many factors that involve whether they're going to try to market something. You notice that almost all of the herbicide-resistant crops that are commercialized are major crops. That's the only way you can get the economic return that you need to invest what's needed to get these products through regulatory approval and the development cost. There are new technologies that are coming online for producing herbicide-resistant crops, such as gene editing, using such technologies as CRISPR-Cas methods. This is an example where CRISPR-Cas has been used to do a 1 codon change in the acetolactate synthase gene in maize. You can see the resistant, the gene-edited variety of the crop on the left there, and it's completely resistant to a sulfonylurea type herbicide. Yeah, this should be chlorsulfuron. So that's one method. The cost of getting these on the market could be quite a bit less than transgenic herbicide-resistant crops, because the regulatory agencies may view these as non-transgenic. Another aspect that impacts all of this is the fact that we haven't had a new molecular target site among commercial herbicides introduced for about 30 years. This slide's slightly out of date, but that top line is still flat. Part of the reason for this is that glyphosate-resistant crops were so successful, they took about 50% of the profit out of the chemical herbicide market, and companies greatly decreased their herbicide discovery efforts because of that. Now we have this tremendous herbicide-resistant weed problem, and companies are beginning to invest more in discovery. Whether we get new modes of action in the near future or not, remains to be seen. There are new concepts for using transgenes for weed management that involve other sites of action that could be used. Here you see a weedy grass grown without phosphorus on the left. In the middle you see the weedy grass planted with tobacco and fertilized with phosphate. On the right you see tobacco transformed with a gene that converts phosphate to phosphite to phosphate, which is a fertilizer, but the grass has not been transformed, and then the tobacco is not affected. In fact, it's fertilized by phosphate, which would be very toxic if it weren't for the transgene. The phosphite is killing or greatly impeding the grass. Now, phosphite was actually used as a herbicide before synthetic herbicides were introduced, but it caused a lot of crop damage. This is a way of eliminating crop damage and using phosphite as a herbicide, in fact, as a herbicide and a fertilizer simultaneously. So there's some things thinking outside the box that could be useful in weed management. Some final thoughts. Glyphosate-resistant crops have been the most successful transgenic crops by far. That's undisputed. They've offered significant environmental advantage over the weed management options that they replace. At least initially they did. The advantages, economic and environmental, of this herbicide transgenic crop option for weed management have eroded due to the evolution of weeds to glyphosate. Herbicide-resistant crops for old herbicides are a stopgap measure to combat herbicide-resistant crops, and herbicides with new modes of action are needed to diversify weed management options. Diversification stops or at least slows evolution of resistance. It may be beneficial to introduce herbicide-resistant crops with resistance to new modes of action, and certainly new non-herbicide-based weed management methods are badly needed to be used either with herbicides or as a lone herbicide management options, and to diversify weed management. And with that, I thank you for listening, and I'm open to questions.